Right, this is going to be our DNA lesson 2. In our last lesson, in lesson 1, we looked at the structure of DNA, that double helix shape. We looked at the nucleotide bases, remember A, T, C, and G, or G and C. We also looked at DNA replication, so the DNA making a whole new strand of itself and copying itself. DNA replication takes place for mitosis and meiosis. Today, we're going to be looking at transcription and translation. And this is how DNA keeps our body functioning throughout the day. So, RNA is a new type of molecule that we're going to talk about. It's also a nucleic acid, and it's what carries the DNA instructions. You see, the DNA cannot leave the nucleus, but RNA, it can leave the nucleus. So this is like a messenger moving, literally a messenger, moving from the DNA inside the nucleus, where it's kept safe and sound, and then moving into the outside scary world of the rest of the cell in order to make proteins that your body needs to do things like digest your food, grow taller, grow your hair, things like that. So this is called the central dogma, and it's basically the theory behind DNA. So there's 3 processes that happen here. There's replication, which copies the DNA. That's in blue right here. And that's what we talked about in our last lesson. Then there's transcription, and this is converting DNA into RNA. This is like you copying your notes from the book to your notes packet. You're just transcribing them, or copying your work from another classmate, which of course I know none of you do. So this is just converting the DNA message into RNA. Finally, we have translation. This is what happens when we take that RNA and turn it into a protein. So this is replication, this is transcription, and this is translation. Right. Now, translation is where we're actually interpreting the message and literally translating it into a different language. So we're translating it from RNA, which is a nucleic acid. We're taking this back to macromolecules. This is a nucleic acid, and it's converting it into amino acids, which, if you remember, amino acids make up proteins. Hopefully you knew that before I said it. So as you can see here, I'll erase my bad handwriting, and you can see that we have replication up here, then transcription taking it into RNA, and then translation making it into a protein. So RNA is the link between DNA and proteins. So DNA stands for deoxyribose nucleic acid. RNA is ribose nucleic acid. We'll see why here in a second. RNA is only one strand. So DNA looks like that and has 2 strands that are connected. RNA is only one strand. And it only has a 5-carbon sugar, so a ribose, not more like the DNA. It does have a phosphate group, just like DNA. And it does contain a nitrogen basis. And it contains the A, the C, and the G. But instead of thymine, it contains something called uracil. So the U, T, becomes U. So when we're doing our practice of coding out DNA to RNA, when we're actually doing our transcription practice, anytime you see an A, it's going to pair up with a U. And anytime, so if this is our DNA, here, I'll do some erasing. If you see on this side, on our DNA, if it's A, T, C, G, for RNA, it would be A would normally pair up with T, but because it's RNA, it's going to pair up with U. T pairs up with A, because we still have an A over here in RNA. C still pairs up with G, and G still pairs up with C. So we just have a slightly different pairing structure. Whereas if this was DNA, you would have a T, A, a G, and a C. So we will have lots of practice with this. You will get very sick of translating and transcribing this information. So again, there's 3 major differences between RNA and DNA. The first one is RNA has a ribose sugar. Second, it has uracil instead of thymine. So it does not have a T, instead it has a U. And it is a single-stranded structure. It has one strand and not two strands like DNA. All right, so there's our nucleotide. So transcription. Transcription is going to copy the sequence of DNA to produce a complementary strand of RNA. So when your teachers tell you to write your notes in your own words, what we are wanting you to do is transcribe. What we are wanting you to do is take the notes that we have, write them down, and then you might have a few changes to make them to where you understand them better, kind of like switching the T in DNA to a U in RNA. So this occurs in the nucleus, just like taking your notes normally occurs in the classroom. And during transcription, an enzyme known as RNA polymerase, it actually, RNA polymerase is an enzyme that goes in, it separates the DNA at just the location of the gene we want. So let's say you drink a glass of milk. That'll be my example for this lesson. If you drink a glass of milk and your body needs to produce the LAC enzyme so you can digest that milk. So the RNA polymerase is going to separate the strand of, or the part of DNA that codes for that LAC protein. It's then going to use one strand as a template, and then new nucleotides are going to come in. And so you'll see a few slides. So again, RNA polymerase, this is an important vocabulary for you to know, and this is what it does. It recognizes this is what it is. So that's the definition of RNA polymerase, is it's enzymes containing proteins, they play a variety role. But what it does is it recognizes where to start transcription, it begins to unwind the DNA, it strings together complementary strands of RNA, and it recognizes the end. So it knows where to start, it knows where to stop, and it knows what to do in between. Right. Transcription is catalyzed. Remember, this means started or began. So it's catalyzed by RNA polymerase. RNA polymerase and other proteins form what we call a transcription complex. This again recognizes the start of the gene right here. So it recognizes the start site. It unwinds the segment that you need. It brings in these new nucleotides to form the strand of RNA. And this whole thing is called the transcription complex. So nucleotides only bond with one strand of the RNA. So see, we're not making a whole new strand. We're just making a little strand right here, you can see in this picture, of RNA. And as we finish, the DNA winds back up, back into its original spot. So once the RNA has the full message it needs, it detaches from the RNA, or the DNA, the RNA detaches. The DNA will wind back up into its original shape, and the RNA will travel along and take its message. So in the history books, you hear about, there's a king in battle and he wants to send a message back to his homeland, and he sends a messenger that runs really fast, which coincidentally is where we got cross-country running from. But this RNA is the original cross-country runner, and it's going to now leave the nucleus and go do a job. It's going to go send a message to make those proteins. And so this is the process of transcription. So you can see another picture of it here. We only have uracil in RNA, and we have thymine in DNA, which we'll get more into that in practices. So the way this works, how does the RNA know where to start? There's something called a promoter in your genes. And this promoter is basically says, okay, start for this gene here. And that's where it binds and it begins transcription. Now, our DNA has been passed down through evolution for billions of years. So we have a lot of DNA that we do not need. So it's just junk DNA now. We don't use it for anything. It's just still there because of evolution. So we have something called introns and exons. We're going to go over this here on our next slide. So we also have mRNA processing. Before the mRNA can send its actual message, it has to edit it a little bit. Kind of like sometimes you get a bunch of notes that you don't actually need. And you just can take those parts and highlight the important parts. So the important parts are called exons, and introns are like intruders, right? So I think of those as like they're intruders into the DNA. You don't actually need them. So what happens is the processing, we take the introns and we remove them, We stitch the exons together, and then that's our actual message. So we just removed a bunch of that nonsense out of our message. Now, transcription does have 3 types of RNA. What we've been talking about so far is messenger RNA. This is what carries the protein. During translation, we're going to talk about ribosomal RNA and transfer RNA. So this is the mRNA. This is what we've been talking about. It's a single strand. You have ribosomal RNA, which, remember, the ribosomes make proteins. This is how they're going to make the proteins. So these form little ribosomes, and we'll get in translation. We're going to get to how tRNA and this ribosomal rRNA work together with the mRNA to actually make proteins. So, translation. During transcription and replication, we're speaking the same language, maybe just 2 different accents, right? So it's both English, but if you have a British accent and an American accent, you can still understand each other, but there might be a few words that are different. That's how replication and transcription are. You just have literally 1 letter difference, T versus U. When we're talking about translation, though, we are literally translating this information into a new language, which is what our proteins speak. So we're transferring it to a new amino acid, which are proteins, macromolecules again. So translation converts mRNA messages into polypeptides, which is the polymer of proteins. And they do this by using codons. Codons is a sequence of 3 nucleotides that codes for an amino acid. So you have lots of letters, C, G, and we're actually going to go ahead because we're an mRNA. We're going to change that T to a U, and then it might be G again, and then C, and then U, and then A, and then C, and then G. It doesn't matter, whatever order these letters are in. So this is your mRNA strand. What we do with our mRNA is we break it into 3-letter groups, right? Each of these 3-letter groups is called a codon. It's basically a DNA word. And each codon codes for something specific. You can see it down here. We have 2 codons, AUG and CUU. Now, these code for amino acids. And each codon matches a specific amino acid. There are 3 stop codons. So 3 different codons code for to stop. 1 codon codes for start, which is this amino acid there. Now, there is a very handy chart. I'm going to show you a different chart on the next page because there are only 22 amino acids. So we could have thousands of different codes or hundreds of different codes, but they only code for 22 different amino acids. So it's pretty easy to figure out where we're getting those amino acids from. So this chart, you can see where this is the first base. So say we have C, C, U, and that's what we're coding for. We can look at our first base is C, our second base is C, and we find the center here where the 2 C's interact. We find CCU, and it codes for proline, right? If we had, let's say, CAU, which is what this example on this page gives us, we look at our first base, which is C, our second base, which is A, our third base, which is U. So you trace C into here, trace A down, and then find CAU. So you're using this chart as kind of like a key. You can also use this circle chart, which I personally like this one better. You start in the center with your first base, then you get your second base here, and then finally your third base are these tiny letters on the outside. So what we see here is we would go A to C, and that gives us this amino acid there. Now the amino acids have long names, as you can see. We often abbreviate them. So valine would be VAL, and it would be abbreviated. But because your test is multiple choice on this, as well as when you see them on the EOC, this will be explained. So it'll either give you the whole word, or it will give you the abbreviation. So the genetic code is the language of mRNA. It's based on the 4 bases. Each 3-letter word is called a codon, and the strands are separated into 3s. So when you do your practice worksheets, remember you're always separating them into groups of 3s. And it makes it way easier when you have a strand like this to actually go in and actually separate your strands into. So if we change the order in which codons are read, it will actually change the protein. For instance, if you change the order of words in a sentence, it can change the meaning of that sentence, right? So if we start here at the C and CGA, we get this set of protein, or this set of amino acids. Those amino acids are then strung together into a protein. But if we start even one letter over, we get 3 very different amino acids, which means we're going to get a very different protein. So it does not matter, and this is extremely important. I know there's a question on your test about this. I don't care if what we're talking about is a stalk of corn, a human being, an earthworm, a dinosaur, or think of an animal or plant or fungi or bacteria. It does not matter what we're talking about. Any living thing, codons code for the same amino acid. So if CGA, if it equals ARG, if it equals this protein in humans, it's going to equal this same protein in corn, the same protein in earthworms, the same protein in dinosaurs, the same protein in chimpanzees. It does not matter. So the codons always code for the same amino acid, right? It's like kind of like translating from English to Spanish. One day, it's not just going to randomly change and this English word is going to equal a different Spanish word, right? They're always going to be the same. Now, amino acids, those little codons together, are going to link to become a protein. So this is the second part of it. So we've coded the mRNA. We know what amino acids they link to. But now we need to actually do the process. So if you have your code of mRNA here, and it has 3 different things. Let's say it's AUG, right? This anticodon, right, comes in and pairs with it. So this is part of that tRNA. So this is going to come right here, and it's going to pair with these. And so the anticodon, what pairs with A? It would be U would be A, and G would be C. So the anticodon would be UAC. This is going to carry the amino acid. Now the tricky part here is this amino acid based on our chart is based on these. This is just kind of like the Google Translate step in between. You don't really need to memorize these letters here. The only letters you really need to know are down here. And again, you don't need to memorize any of this. The charts will be provided for you on the test. The amino acid is brought to this mRNA, but it's brought by the transfer RNA. So the transfer RNA is kind of like an Uber for the amino acids. It connects in and it moves on. So ribosomes have 2 subunits. So here you would get your mRNA. So here would be your AUC. Here you would get your binding sites with your anticodon, so UAG. And then this is where the amino acid would link in here with the tRNA. So here's our process. You have your strand of mRNA down here. That's like our conveyor belt that's coming through. Then you have your ribosome, right? And your tRNA. Your tRNA is coming in. It's attaching to the mRNA with the anticodon, so it's attaching. So this way, only the correct things will attach to each other. And it's bringing the amino acid. As it moves through, the amino acids link together and become a long chain of polypeptides. And those amino acids will eventually protein. So the ribosomes help form the peptide bond between the amino acids, and the ribosome pulls the mRNA strands the length of the codon. So you can see down here the same process is happening. We have the start codon, the transfer RNA coming in, it's bringing the amino acid, and then the new one's coming in and they connect together. All right, so it's very much like an assembly line in a factory. Now, the empty tRNA molecule, the tRNA, is now going to exit the ribosome. It no longer has an amino acid on there. It did its job. It dropped it off. And then the next exposed codon, the new tRNA molecule, will bind to it. Once the stop, so this process continues, so you can see you get a long chain of amino acids. Once the stop codon is reached, this process will stop. That part of the ribosome, or this ribosome, will break off of the rRNA. The ribosome will break away, and the protein is made. And we're done. Now this protein is going to go to the endoplasmic reticulum. It's going to fold it all up nice and pretty and send it on about its way to do its job. So what happens when this goes wrong? So up for now, everything's been working great. But if there is a mutation in the gene that was not caught early on by those checkpoints during the interphase of mitosis or meiosis, if there's a mutation, this can actually, what it is, the change in the organism's DNA. There are lots of types of mutations that can occur during replication and then not be caught. So an example is a point mutation, and this just substitutes one nucleotide for another. So for instance, originally you had this chain here, but then this changed from a C and it mutated accidentally into an A. So now that A is going to code for a U instead of a G like we saw here. Now it's going to code for a U, and now it's going to code for a completely new protein in your, or completely new amino acid in your chain of proteins. This is called a point mutation, and it's just at one location. Another type of mutation is a frame shift mutation, and this is basically when a nucleotide is erased or is added in. So it shifts everything down, or it deletes it and shifts everything forward. And this makes the DNA chain of amino acids mess up. But a point mutation, only one of these amino acids is going to mess up, and then they'll continue on like normal. During a frame shift mutation, though, everything from the mutation on is now going to be messed up because every three-letter word or codon is read differently. Next, we have chromosomal mutations, and we talked a little bit about these when we were in meiosis. Chromosomal mutations affect lots of genes, and this happens with uneven crossing over. So during meiosis, we talked about how the telomeres, or the lengths of sister chromatids, will cross over and exchange some DNA. Normally, when this happens, the chromosomes are lined up equally. I drew that X weird. The chromosomes are lined up equally, so when they switch the same amount of DNA. In this sense, the green chromosome here, this homologous pair, is giving less DNA than the blue homologous pair. So now we have less DNA on this chromosome and more on this chromosome. We can also have translocation from non-homologous chromosomes. For instance, instead of these two chromosomes crossing over and these two crossing over like they were supposed to, these two chromosomes crossed over. And so this causes a major problem because you have DNA in completely the wrong spot. Chromosomal mutations tend to have a really big effect, like as in non-survival of the organism. Or if the organism does survive, it's going to be have some major problems one way or another. Now, some of these problems are very easy to live with, some are not. Gene mutations can cause a change in phenotype. Remember, phenotype is our physical expression, or it can be internal expression, like not being able to produce a certain enzyme. So this might cause a premature stop codon. In other words, you don't produce enough of whatever enzyme we're talking about. The mutation may change the protein shape or the active site, so it may change the way the protein is made, and it might also change gene regulation. So for instance, a gene mutation might cause a certain buildup in blood vessels and arteries versus not. And so this is where we have, genetic high blood pressure and things like that. So things that can be passed along is that the genes are regulating and the proteins are being made to cause an issue in the body. Some gene mutations do not affect phenotype at all. A mutation might be silent. Kind of like if you've typed up a paper and you made a typo, if nobody notices, no big deal, right? So this could maybe be because it's in a non-coding region or a part of your paper that nobody read. Or it just might not affect the protein folding. Remember, several different codons can equal the same amino acid. So even if it's mutated, if it's still coding or if it's still translating into the same amino acid, then it doesn't matter. Mutations in body cells do not affect offspring. For instance, if there is a mutation that happens during your lifetime to, let's say, a skin cell, that mutation is not going to be passed on to your offspring. Mutations in sex cells during meiosis can be harmful or beneficial to the offspring. So this is where we get into evolution. So mutations that happen during mitosis, those are safe. They are not harmful. Mutations that happen during meiosis are bad. Those can be harmful or they could be beneficial, but they're going to have an effect. So natural selection often removes mutant alleles if they're less adaptive. So for instance, if a polar bear was born with really dark fur, it's probably going to not survive very long against that white background of snow. Versus a lizard that's in the jungle, if it's born with bright red scales, it's going to stand out against those green leaves, and it's probably going to be eaten pretty quickly. So natural selection removes those mutant alleles. Now, if those mutant alleles are adaptive, let's say a lizard's born the exact same color as the leaf, this is where natural selection will work in their favor. Mutations can be caused by different things. You can have replication errors, like during mitosis, and this is just a random chance. It just could happen, like a typo. You can also have things that cause mutation, like UV rays from the sunlight, chemicals such as asbestos and other things can cause mutations. Some cancer drugs use this to kill cancer cells. So they use properties that just kill mutations in the DNA. So this is some new cancer research there. So lots of mutations can happen for different reasons. And so normally the two of these things work together. This is why we say cancer is both genetic and environmental.